Hi everybody, Mrs. Mancuso here. I am going over the week 7 FRQs that I gave you, and this is question 4, probably my favorite of the 4. So it talks about Drosophila, which are fruit flies. So species of fruit flies frequently used by researchers in genetic studies. Members of this species have 2 of each of the 4 different chromosomes. The sex chromosomes, flies have X's and Y's, and 3 autosomes, chromosomes 2, 3, and 4. Researchers studying fruit flies conducted genetic crosses to investigate a particular X-linked recessive trait encoded by a single gene. Affected flies have the trait. Okay, so affected flies are going to be, I'm going to call it X little a. And so if it is an affected fly and it's a female, it's going to be X little a, X little a. And if it's an affected fly and it's a male, it's going to be X little a, Y. Okay, so then they give us this data chart, which I think is very small and difficult to read. So for each of the questions, I blew it up a little bit larger. So they did 4 different crosses. They tell you what mom's phenotype is. They don't tell you her genotype. They just tell you, does she have it or does she not have the trait? And then they give you the phenotype of the male. So again, but for dad, whatever his phenotype is, that's directly due to his genes because he only has one X. So because he only has one X, if he is affected, he's got the little a. And if he is unaffected, he has the big A. And then it tells you the percent of the total population of their offspring, and then their offspring that are males, the percent that have it, and percent of females that have it. So that's how you read that chart. All right, let's go to the first question. Identify the genotypes. So that means they want me to give the letters of the male and the female flies used. So the male is going to be XY. The female is going to be XX, used in cross 2. So here's cross 2. The female is affected and the male is unaffected. So the female, since she is affected, she has to be X little a, X little a. It's the only way that a female would express a sex-linked trait is if both of her X's had the recessive gene. For the male, he is unaffected, which means he's X big A, Y. Now that's all they asked us to do. The verb identify says tell me the parents. So I would write this is the mom, or this is the female fly in the cross, and this is the dad, this is the male fly in the cross. If you have time and want to go back and check all of your work, you can. All of this data makes a lot of sense. Look at this one. Percentage of male offspring. All of the boys have the trait. They have the phenotype. That's because mom could only give away X little a. To the boys, dad gives the Y. So all of the males are going to have it. None of the girls are going to, because all of the girls got the X from dad, which is X big A, and the other X from mom, which is X little a. So all of the daughters are carriers, but none of them actually have the phenotype. All right, let's go on to the next question. Identify the cross. So another identify verb. All I have to do is identify if it's cross number 1, 2, 3, or 4, in which the female parent was most likely heterozygous. So I want to figure out where the girl is X big A, X little a. So the first thing I'm going to do here is I'm going to cross off cross 4 and cross 2, because 2 and 4 both show me that mom is affected. If mom is affected, she has to be X little a, X little a. So it can't be cross 2 and it can't be cross 4. So if the mom is heterozygous, X big A, X little a, and let's just look at cross 3. So if mom is unaffected and she's X big A, X little a, and dad is affected, that would mean he's X little a, Y. And I would expect then that half of the daughters are going to have the trait. So I go over here to the females and none of the females are getting that trait. So that's not possible. So can't be cross number 1. So my answer is cross 3. So let's look at it. So if my female is X big A, X little a, and dad is unaffected, so dad is X big A Y. If I did the cross, so I go down here, X big A, X little a, X big A, Y. If I do the cross, this is a girl and she's unaffected. This is a girl and she's unaffected. This is a boy and he's unaffected. This is a boy and he is affected. So of the total population, I would expect 25%. Yes, that's what you see right there. Of the males, half of them. Yes. Of the females, 0. Yes. So my answer is definitely cross 3. All right, on to the next one. The researchers hypothesized that crossing any unaffected female and affected male will result in 0 chance of producing an affected male offspring. Evaluate the validity of the hypothesis. So when they say evaluate the validity, I want to know, are they correct or not? That's what it's asking me. Is this valid or not? So if I have an unaffected female, that unaffected female could be X big A, or X big A, X little a. Both of those females would be unaffected. And I'm going to cross them with an affected male. An affected male is X little a Y. And it says 0% chance of having an affected male. Well, the males get the Y from dad and they get the X from mom. So if mom was X big A, X little a, half of the males would be affected. So the question said, evaluate the validity of a hypothesis. I would say that the hypothesis is not valid. Because the female fly can be X big A, or X big A little a. And X big A, X little a would lead to 50% of the male offspring being affected. Okay, on to the last part of this question. Explain how the results exclude the possibility that the trait is encoded by a mitochondrial gene. Okay, how do these results say that it's not possible that it's a mitochondrial gene? Okay, so first of all, I want to go back to mitochondrial DNA. So we know that if this was my prokaryotic cell, the way that it became a eukaryotic cell was through endosymbiosis. So endosymbiosis showed us that originally when this cell took in another cell, and instead of destroying it like it normally would for food, it kept it inside. That's how you got a eukaryotic cell. So this mitochondria used to actually be its own separate identity, its own separate cell. That mitochondria has its own DNA inside of it. Now let's talk about how mitochondria are passed in fertilization. So here is my ovum or my egg, and it has a haploid nucleus, and it definitely has mitochondria. So here it is, and I'll show it in green. The mitochondria have their own DNA. Obviously, the nucleus has DNA too, like all the experiments that we were just doing. Now I'm going to show, oh, I don't want an arrow here. Okay, now I'm going to show the sperm. So here's the sperm. The haploid nucleus is right here, and there is a mid-piece region, and this is where the mitochondria are found, in this mid-piece region, giving it energy for the tail to move. When fertilization happens, the N goes in, and then it breaks off. This whole section, including the mitochondria and the tail, never enters the egg. So my zygote is my original ovum cell with all the original ovum mitochondria and the mitochondrial DNA. And now it has a diploid nucleus. But all of the mitochondria came from mom. So I needed to do all of that explanation to go back to the question and say, all right, the question said, explain how the results exclude the possibility that the trait is encoded by a mitochondrial gene. All right, if the trait was passed on through mitochondrial DNA, all offspring would have the same as the mother. Mitochondrial DNA is only passed from mom to the offspring. That is not true in the data above. So that is showing us this is normal nuclear DNA, because it is showing that it can be passed on from the parent, father or mother, to the offspring. So that means it has to be nuclear DNA, not mitochondrial DNA. If all of the children were exactly the same as the mothers, then it would show that it could be mitochondrial DNA if it did not matter what the dad's nuclear DNA was. All right, and that is the end of question 4.